for the hour and you should be okay.
35. Oh, do we have all the kids again? I don't know how. All right, yeah, and then also we're gonna just.
Oh, someone named Miss Eggers.
We're just gonna. Just say hi.
What?
Say hi.
No, she's been watching all day. She doesn't need me to call her out.
His mom's watching because she's a teacher.
My mom is watching also.
His
My
mom.
my mom's a teacher,
Maybe your
so
mom's
she's
watching
watching
too.
right now.
Moms care. Moms are
Probably showing it to her entire middle school classroom right now.
my mom. I'll just put my hi mom out there too.
Okay, it's too much. Let's
All right, time to go. Cool.
All right. So, hello everyone. Thank you for joining us today. Today is a particularly special day. Even though it is officially tomorrow, today we are observing the International Day of Women and Girls in Science. And so next up, we have Neil Balchan, who will be presenting on snakes. So,
yay!
All right. Well, thanks for having me, guys. I'm gonna try to screen share, and hopefully I don't destroy whatever we have going on here. Let me see. Come on. All right. I think we're looking at a PowerPoint presentation. Can somebody give me a thumbs up if that's what we're looking at? All right. Okay. I think we're good to go. I'm just trying to hide this little screen. All right. Ben is super scared of parents, but their parents are making them watch this. All right. Well, thank you, Ben, for joining us today. Okay. So I assume we're good to go. Can I just get a thumbs up from somebody if we or just something in the Q&A?
You're all good. You're good.
Okay, we're good. Okay, cool. All right. I'm always unsure if I'm showing a screen or not. Okay, cool. So thanks so much for having me, guys. My name is Neil Balchan. I'm a PhD student at Oklahoma State University currently. And I just want to give Hadley Copeland a shout out. She's the reason I'm here today. And I'm very appreciative always of the opportunity to kind of ramble about snakes. So if at any point you have a question, some of this stuff can get kind of heavy and dry. So if you do have a question, feel free to just throw it in the chat. I'll try to check up on that kind of as we go through. But with that said, I do anticipate we'll have lots of time at the end. And we can definitely have a bit of a back and forth there. All right, cool. So I'll dig right into it. So generally, I talk a lot about really weird specific things, and that's kind of what I'm going to do today. But I want to kind of build this precedent that while we do know some things about snakes, we actually don't know very much about snakes at all. And I think that's kind of what's been the driving thing that's kind of kept me going on this weird little trajectory in science. All right, so I guess just a quick primer to snakes. So there's around 4,000 species of snakes. In fact, this number just reached 4,000 quite recently. And kind of across that 4,000, there's this incredible diversity. So I guess if we just run through these images, there's a sidewinding rattlesnake, which is this beautiful little rattlesnake that kind of moves through the sand in a brilliant little way. And this is a viper, right? So then we have a group of snakes called the elapids. And you can see this middle image here is a coral snake. So, red touch yellow, kill a fellow. This is a diverse radiation of snakes that are, horribly toxic. They're spread all over the world, and they're just wonderful. We have snakes called colubrids or colubroids, I guess we're using now more as a term. And this includes things like the garter snakes. Most of your kind of common snakes fall into this basket, right? Of course, we have the giant constrictors. These are two families. So the green anaconda, kind of in the bottom corner there, and then the Burmese python right beside it. These are examples of boas and pythons. And while most of these, or most of the charismatic ones at least, are huge, it's important to remember that there's some tiny boas and pythons too. And then just kind of a weird little tidbit here, this guy in the other bottom corner is called a shield tail. And this is a really cool radiation of snakes that are present really throughout the Indian subcontinent. And they have this hard actual shield on the end of their tail. All right, so Cadence asked me if I'm going to show a cobra. Unfortunately, I'm going to show a lot of cobras, so be ready for that. And then why do most snakes have a pattern? Okay, really good question. Super variable, right? Patterns might be used for kind of camouflage. Maybe if you're red and yellow, you want to be startling. You want to kind of scare predators away. So there's a bunch of different reasons that a snake might have a pattern. All right, so again, diverse radiation of snakes. And I think this is what's captivated me from the get-go, right? I think I've got my mom in the audience, so thank you for tuning in today. But as a young kid, I was weird. I was obsessed with snakes, right? And this is a picture of me at a young age, kind of just messing around with garter snakes with my family. And I think, you know, at some level, maybe my family would have hoped that I could shake this off and that I'd be a normal kid and I'd be playing basketball and, you know, doing normal kid stuff. That didn't go away. And I think I just became more and more obsessed with this kind of diverse radiation of snakes and trying to answer some of these questions that we don't have an answer to. And again, this is baby Neil, but that took big Neil all over the world. And I've spent a lot of my free time just trying to learn about snakes, even outside of the classroom or outside of the lab. And this past summer, I was really fortunate to spend a lot of time in Southeast Asia. So the image on kind of the left side of the screen there, that's in some remote mountain in Laos, actually. And that's a three-horned pit viper, this kind of dragon of a snake, if you will, that lives in this really, really rough terrain. On the right is a temple viper from Thailand, a super beautiful snake. So I think this obsession for snakes kind of doesn't really know bounds, unfortunately, but it's also kept me really motivated to just learn more and kind of build our knowledge. All right, so as we heard pretty early on, we're really fortunate to be kind of celebrating, I guess, a day early here, but International Day of Women and Girls in Science. And there's a lot of women that I've been really, really fortunate to kind of have as mentors and as friends in this field. And in fact, some of the really pivotal kind of knowledge that's come out of here has been generated from some of the women that I have on screen. So I just want to highlight the work of Dr. Anita Malhotra, which has been really fascinating. Anita's contributed a lot about what we know about Indian snakes in particular, and I guess snakes more broadly even. Dr. Elda Sanchez, based at Texas A&M Kingsville, is kind of a pioneering force of venoms. A lot of what we've learned about venoms comes out of her group, and I've been really fortunate to have her as a mentor over the years. I'm currently working in the lab of Dr. Gwyn Wogan here at Oklahoma State. Gwyn is a giant in Southeast Asian herpetology. She's really taught us a ton about how snakes and amphibians also work in Southeast Asia. And then Dr. Kate Jackson has been a pioneer of African snakes. She's launched a bunch of really, really great expeditions into the Congo and kind of provided knowledge that we wouldn't have were it not for her trips. So keep these people in mind. And if you want to kind of go down a rabbit hole of their work, I'd really encourage you to check them out. All right, so let's kind of dig into what today's talk looks like. So today's going to be a bit of a mishmash, but I hope you're able to follow along for the ride. So I'm going to start off on this topic that I call biochemical ecology of venomous snakes. And then I'll kind of segue into what I'm working on now. And that's snake biodiversity, snake venom, and then kind of human health and how that all relates back to us. All right, so what is biochemical ecology? Well, I'm sure a lot of you are familiar with regular ecology, and all ecology is, right, are the biotic or the organism interactions that are happening in a habitat. So you could think about a lion eating a gazelle, or, you know, really any other two wild animals or plants interacting with each other. Now, biochemical ecology puts a weird twist on that because we're looking at the same kind of interactions, but we're looking at other things that factor in the biochemical sense. So poison, venom, and just toxins more broadly. So we could think about the monarch butterfly, right? Whose bright orange colors tell birds, hey, I'm toxic, don't eat me. Poison dart frogs do the same. So this bright orange dart frog is signaling to predators, hey, I'm toxic, don't eat me. And then on the right, we have this interaction here where a garter snake and a California newt, the garter snake is eating the California newt, and this newt is incredibly toxic. If you lick this newt, you would die. But the garter snake is toxin resistant. So it's evolved a mechanism to get around that toxin And this is where things get really cool, because we have this system in the natural world where toxic things are getting eaten by things that are immune to toxins. And that's what creates this biochemical ecology. All right, so I like to generally start things off with quotes because I like quotes a lot. And this quote is going to get a lot of mileage today, so bear with me. But this quote came from A.F. Scutch, who's an American ornithologist, so he really likes birds. And he said that the serpent crams itself with animal life that is often warm and vibrant to prolong an existence in which we can detect no joy and no emotion. And then he went on to say further that it reveals the depths to which evolution can sink. Now, as a snake lover, right, I don't agree with that, but Scutch came up with this seeing this type of interaction, right? Seeing a viper maybe catch a hummingbird or something like this. And it bothered him, it triggered him, right? And to his credit, that's fine. But I take a little bit of offense to his final statement of it revealing the depths to which evolution can sink. And I think nothing could be further than the truth. All right, so venoms. What are venoms? So venoms are really this beautiful golden or amber liquid that comes out of a snake. But when we think about them in a little bit more depth, they're a complex mixture. They're a cocktail, if you want to put it that way. And they're made up of different things. So proteins, peptides, and enzymes, and maybe some other things as well. But venoms do a bunch of different things, right? We think about venoms being dangerous, but for a snake, they're a tool, right? They're a functional trait. So venoms can be used for digestion. See that in a lot of rattlesnakes. Venoms can be used for defense. And somebody wanted to see a cobra. Here's a cobra. This is a spitting cobra, right? These snakes can spray their venom defensively. But I think the most important thing, and the thing that this talk is going to really be hinged on, is this idea of prey acquisition. So can your venom help you in A, capturing and killing a prey? And then potentially also B, can it help you in eating that prey? And that kind of goes back to digestion. All right, so let's start off by understanding what snakes eat. So let's think about a rattlesnake. I'm gonna give you guys 30 seconds in the chat. Just go ahead and write whatever you think a rattlesnake eats. I would love to hear what you come up with. So what does a rattlesnake eat? All right, I'm seeing mice, I'm seeing mouse, I'm seeing rats. Snakes do not eat lettuce, Milan. I'm very sorry. There are no vegetarian snakes. All right, so a lot of mouse and rat, right? And those are really, really good answers. So if you think about a snake maybe living, in captivity in a lab or something like that, mice and rats are what we feed it. If you think about a snake in the wild, right, mice and rats are probably what it's eating. So a lot of rattlesnake diets are composed of this, right? Mice, rats, you might even get the occasional bird in there. But again, warm-blooded prey, things that are common on the landscape. But I want to throw a wrench in it because we don't know what snakes eat. No one's out there watching a snake 24 hours a day, watching what it eats. And because of that, there's things that snakes eat that we've never even thought of. And one of those things is centipede. So I'll give you guys this video to watch here. This is a rattlesnake from southern Colorado. It's called a Massasauga, and it actually can eat centipede. You can see it's doing a couple weird things, right? It's raising its body. It's behaving very carefully. So it has kind of this innate compass of how to hunt a centipede, and it should bite it in a moment, but we'll give it a second here. So spit in the centipede and notice it let it go immediately, right? Because centipedes are dangerous. They have a strong bite, they have venom. Really, really weird scenario here. We wouldn't think a rattlesnake should be eating a centipede, but it is. So the only point that I wanted to make with that video is that a lot of the things we have in our heads are assumptions, right? And what we don't know is often a lot more interesting than what we do know. So that's all the centipede talk I've got for today, but I just wanted to bring that up. Oh, and it's starting again. Okay, so let's kind of think of the most basic scenario, right? We have maybe a diamondback rattlesnake, he's coiled up under a bush, and a deer mouse walks by. So deer mouse walks by, rattlesnake does one of those, boom, our mouse has now been envenomated, right? It's been bitten, there's venom coursing through its veins. So ordinarily, a mouse could try to run away, right? And that probably won't work in this scenario. The mouse isn't going to make it. The mouse could try to hide. Again, probably not going to get him far. Or that mouse could do something that's really, really cool. And he could actually turn off that venom that's inside of his body. He could use something we call an endogenous mechanism to catch that venom and to put it away and to kind of get it away from what it's going to do, right? Which is break his body apart. And I think this is our light bulb moment, right? Because if we think about Scutch from a moment ago, he said something mean. He said the depths to which evolution can sink. And that's not at all what's happening, because we've opened up this beautiful evolutionary can of worms. And we now have a scenario that evolution can act on, and it can build on, and it can make some really, really, really cool things happen. All right. And what this is, it's actually described in the literature. This kind of scenario is called the Red Queen hypothesis. And they're stealing a lot of ideas from Alice in Wonderland here, which is why we've got that little figure. But it's this idea that organisms in the landscape, in the wild, so again, right, lion and gazelle maybe, these things are interacting with each other. So if I'm eating something and something is, getting eaten by me, we're interacting pretty closely. And because of that, evolution needs to work on both of us together. So every time the lion takes a step in evolution, if the gazelle wants to stay alive, the gazelle needs to take a step in evolution. And that's how we get this kind of teeter-totter dynamic between partners. All right, so if we think of actual examples of this, it sounds like a great idea, but does it actually happen? It does actually happen, right? And one example of that is in these tiny little crustaceans that live in water. So think of like a crab, but they're built a little bit differently. And they're called Daphnia. And they get parasites. And these parasites have to co-evolve with the Daphnia. So again, this is an example of that teeter-totter playing out. We also have garter snakes and newts. You're good to come on in. We have garter snakes and newts. So like I talked about a moment ago, right? These garter snakes are eating newts. Newts are highly toxic. And garter snakes are toxin resistant. So we get this teeter-totter playing out again, right? Where the newt becomes more and more and more toxic, and the garter snake becomes more and more and more resistant to that toxin. So bear in mind, this happens, it's real. So what about venomous snakes and rodents? Well, that's kind of what kept me up at night. And it actually kept me up at night all through my master's, and it still keeps me up at night. So that's what I played with. That's what I wanted to learn about. So what do we know about this system? Well, ground squirrels. Ground squirrels are a beautiful example. And people have been looking at ground squirrels forever. So this cute little critter, actually, he's called a California ground squirrel, is very resistant to rattlesnake venom, to the Pacific rattlesnake venom. When I say very resistant, I mean he's very resistant, but he's not fully resistant, right? And that's why you can see in this image, he's actually being eaten by a rattlesnake. So sometimes he's good to go. He gets a thumbs up at most of the time. But it's important to remember that this is a dynamic situation, right? So sometimes a rattlesnake will eat him. So neat scenario. But then we see the opposite in some cases, and this kind of raises a lot of question marks. So let's think about a ground squirrel in South Africa. This is the Cape ground squirrel, and this thing is the happy meal of the savannah. Everything eats him. So the Cape cobra, there's another cobra for whoever wanted to see cobras, the puff adder, there's a bunch of snakes that eat him, and he doesn't have any resistance, which is insane. You're interacting with all these snakes and you're not resistant. So weird, right? Why is this guy not able to detoxify any of that venom? So this slide goes into a lot of depth. It's not super important, but I threw it in here just for a little bit of background. So this figure on the top here, this squiggly line that you can see on that bar graph, really just represents all the different things in a venom, right? So venoms are cocktails like we talked about. They have a bunch of different things in them. And these question marks below really just speak to the idea that we don't know how anything works. So BPPs, for example, one compound, we have no idea how a rodent can resist BPPs. There's so much that we don't know, but we do have a general idea of how resistance works. And that's what this bottom figure is here. So if we think about this being inside of a rodent, right, this is all blood in a rodent. These are some different pathways we have that a rodent could actually use to turn off venom. So again, not important for today, but I really just want to emphasize that we don't really know a whole lot. All right, so I wanted to look at this where I lived, which was at the time Colorado. So I did that using two study sites, one in northern Colorado and one in southern Colorado. I lived right by that number one up in Greeley, great little town. And I looked at two different snakes. So I looked at the prairie rattlesnake, which lived at both of these places, both number one and number two. It's a nice, big, large-bodied rattlesnake. And I also looked at the desert Massasauga, which was that same rattlesnake we saw eating the centipede a minute ago. So he lives only at spot number two. He does not live up north at spot number one, and he's small. He's a petite little guy. So different snake entirely, right? And then we picked kind of an array of rodents to work on. So we had deer mice, we had house mice, we had a really cool creature called a grasshopper mouse, and we had kangaroo rats, a ton of different rodents, and these lived everywhere. So these are at both site one and site two. And I wanted to know how resistant these rodents were just in general, right? How resistant are you when you have two different rattlesnakes that are trying to eat you? So I think before we kind of make any big conclusions, we need an idea of what's going on, right? So who's eating who? So desert Massasaugas are little, snakes only have such a big mouth, right? So this desert Massasauga is probably only eating small rodents like the house mouse and the deer mouse. Whereas the prairie rattlesnake is kind of a big guy. These can even eat rabbits and things like prairie dogs. So he's probably able to consume all of these rodents. So to throw one more wrench in the scenario, because it's always more complicated than we think, venom is variable in the prairie rattlesnake, right? And snakes at the northern site, at site 1, have what we call a high myotoxin venom. So myotoxin is this compound that paralyzes things. It doesn't do a great job at breaking them apart, but if you're a mouse and you get bitten with myotoxin, you're going to just drop dead immediately. Whereas the snakes in the south have what we call a high SVMP venom phenotype. SVMP will also be called metalloprotease, and this is something that tears things apart. So if you get this in you as a mouse, it's going to cause a lot of bleeding, your muscles are going to break down, your tissue is going to break down. So it's a slower death. But when you do die, you're mush, right? You've melted from the inside out. And what that means is that these northern venoms are more toxic. So they're about twice as toxic to mice as these southern venoms are. So kind of a cool prediction to just put out from the start. So as a good scientist, I went ahead and I kind of jotted down some ideas. So I think that venom resistance, right, should probably correspond to predation frequency, or how often you're being eaten. So in short, right, if you're a rodent and you're too large to be eaten by that desert Massasauga, you probably won't be very resistant. But then on the flip side, if you're a rodent, you're going to be eaten by a prairie rattlesnake. So at some level, you're probably going to be resistant. And then I think that venom resistance will probably depend on geography. And all I mean by that is that if you're a rodent and a snake and you live at the same spot, you guys should have more of a resistance dynamic than if you're a rodent from one spot and a snake from a different spot, right? Because you guys don't interact in the wild at all. So those were the kind of the ideas that I ran with moving forward. So what I did is I harnessed my wonderful army of undergraduate students, and these guys were snake wranglers. You can see Parker here. She's holding just a nice chunk of a prairie rattlesnake. So we went ahead and we collected snakes. We took venom from them and we brought that back to the lab. And then we also trapped a bunch of rodents. So here's a cotton rat you can see in a trap, and we brought those back to the lab. And we used 2 different tests to try to get an idea of what venom resistance means. So it's not very important that you understand what these tests are. I'm just going over them so you might be able to kind of be familiar with what I'm going to talk about. So one test is called the LD50 assay. And what this is you basically take a little bit of venom and you inject it into an actual mouse, into a living mouse or a living kangaroo rat, and you try to find the amount of venom that's going to kill half of that mouse population. So this gives us a good idea of general toxicity of a venom. Okay, so we'll see these numbers a lot. You basically get a number like 1, 2, 3, and we can compare these numbers. We use another test, and this test uses blood. So we take a little bit of blood from a rodent, and we basically put that in a tube, we add some venom to that tube, and we shake it up. And then what we're gonna do is we're gonna measure how much that venom broke down that blood, right? So if we have a really high breakdown of blood, we're gonna get a really high number, and vice versa, right? If blood isn't broken down, we get a low number. So this tells us how kind of protective or inhibitory that blood is against snake venom. So 2 different ways to kind of get at a similar answer. So I'm going to show you a bunch of graphs and a bunch of numbers. Don't worry at all about the numbers or the graphs. Worry about the idea. So these are LD50 curves, right? So again, whole mouse injected with venom. And the gist here, what we see is actually really interesting. It's that these Weld County prairie rattlesnakes, right? So the rattlesnake from the north, his venom is actually more toxic to a mouse that he co-occurs with than to a mouse that he doesn't co-occur with. So that completely goes against our prediction. And what it means is that if we think about that teeter-totter from a moment ago, right? That right now we're actually looking at a time where the snake is winning. The snake is higher up on the teeter-totter. So his venom is more toxic to his co-occurring mouse than to his mouse who doesn't co-occur with him. So really, really cool. So the snake is maybe winning the race in Weld County. But that's just for now. Maybe in 100 years, maybe the mouse will be back on top. So kind of a neat little result. And there's just red boxes showing the numbers that matter. But again, don't worry about numbers today. All right, so deer mice, once again, this is that test where we put the blood and the venom together and shake it all up. And the gist is that these percentages I'm showing you are very, very, very low. So there really isn't much going on with the blood of deer mice in general that's actually shutting down snake venom. We can see in that one example in the top right, we get 31%, I think it is. So that's a little bit higher. But again, right, still pretty low across the board. All right, house mice. So house mice are a weird scenario in that they're not native to the US. They didn't evolve here. They don't belong here really. But Europeans brought house mice to this continent when they came to North America. So mice are kind of a new arrival on the landscape. And what we wanted to see was, do these wild house mice, are they any more resistant to rattlesnake venom than like a lab mouse would be? So we went ahead and we ran that same LD50 assay, and we found that these feral or these wild mice are actually not any more resistant to prairie rattlesnake venom than a domestic mouse is. So in, I don't know, maybe the 1,000 years that house mice have been here, they haven't evolved any defense mechanism to snake venom. So kind of a weird result almost. All right, Plains pocket mouse. This is a cool little rodent. He's tiny. He's about the size of a marshmallow. And because of that, we thought this would be a really good food item for that Massasauga, right? That smaller rattlesnake. So we took mice from the northern field site where there is no Massasauga, and we took mice from the southern field site where there is Massasauga. And we wanted to see if these mice had any difference in their ability to inhibit that Massasauga venom. And we don't see any difference. So for whatever reason, this mouse is probably eaten by Massasaugas, even with that pressure, even with that predator nearby, it hasn't evolved any resistance. So kind of weird. Wood rats, we played with wood rats a little bit, and there's a lot of literature on wood rats. We basically found the same thing. Wood rats are very good at turning off venom. But a really interesting result is that we found that wood rats could not at all turn off venom from that desert Massasauga, that really small little snake, right? And intuitively, that kind of makes sense, right? If you're a small snake and there's a big rat, you're not going to eat that big rat. But to turn off 0% of that venom is a super interesting result, because you would think that if it can inhibit another venom, maybe it should also inhibit this venom. I don't know, jury's out, but 0% inhibition is really neat. All right, and this is where things start to get kind of hairy in the good way, because that northern grasshopper mouse, that critter I talked about a little bit earlier, no one's ever really played with it, so we didn't know what to expect. But when we did that test with the blood, here's what we found. We found really high inhibition or ability to kind of turn off venom across the board. So these mice look to be pretty resistant. But again, blood doesn't really tell you the whole story. So we went ahead and did an LD50, which is where we took these mice and we injected them with venom. And we found that grasshopper mice are bulletproof. I mean, these things are running the marathon when it comes to venom. So grasshopper mice, we got an LD50 score of 127 milligrams per kilogram of venom, right? If we think back to lab mice can take 1.25 milligrams per kilogram of venom. So these guys are literally 100 times that. I mean, they're phenomenal in inhibiting this venom. They can turn off rattlesnake venom like no tomorrow. And the bit that gets really, really weird is that the venom we used was this Weld County venom, right? This is myotoxin. So this is stuff that should be crazy, crazy strong. It should be knocking mice down like that, right? But for whatever reason, these grasshopper mice don't want to die. And they seem to just shut down rattlesnake venom. So I really want to play with this more. We also looked at kangaroo rats, and we can see a similar result. These guys are also bulletproof. Same deal, right? Super, super bulletproof rodent. So I really want to play with this more. This is kind of the height of the data we have right now, but the take home here, kangaroo rats and grasshopper mice will not let a rattlesnake, show them how to die type of thing. So future work. So there's a bunch of stuff I want to do, but I think in brief, the most interesting thing here that I really care about and really want to dig into is isolating what proteins these are that are floating around in the blood, right? So in the blood of these mice, what's actually floating around in there that's doing it? So I'm not a very good lab rat, if you could tell. I like snakes too much to spend my whole day in the lab. But we played with this and we used a technique called affinity chromatography. So basically, we take these little tiny, small, very small beads and we coat them in rattlesnake venom. And then we go ahead and we run rodent blood through that. And what you end up getting is a bunch of proteins that stick to the beads. And in theory, in a perfect world, any protein that sticks to that bead is a protein that recognizes rattlesnake venom, right? And probably a protein that catches that venom and turns that venom off. So we went ahead and we ran the data from that. We ran that on a gel. You can see a gel in the bottom right. And we found something. I'm not really sure what we found, but it is something. So if you look at this gel, you can see some really dark purple kind of blotches in 3 of those rows there. And those purple blotches are protein. And that's the protein we captured. So the jury is out on what that protein is. I have no idea. I am not really a protein scientist, but I would love to get somebody to dig in that a little bit more, because I think if we can really lock down what these proteins are, we can understand this question at a much deeper depth. And again, not knowing stuff, right? I want to know. We have to know. So I would love to dig into that more. So big picture, why does any of this matter? Well, again, so snake bites a mouse and a mouse maybe has a protein in it. Maybe we could relay this back, right? So what if a snake bit a person? Wouldn't it be ideal if a person had a protein in it that could do the same thing? Anyway, so kind of a looming thought, but I'll leave that there for now. All right, I'll give you guys 30 more seconds. Let's shift focus. What eats a snake? Can anyone go ahead and throw in the chat what eats a snake? I would love to hear your ideas. What eats a snake? So if I'm a rattlesnake and I'm squiggling around on a rock, what eats me? All right, I got some stuff. I got birds, I got IDK. IDK is a great answer because IDK all the time. Birds, a leopard, probably a leopard, a lion, mongoose, crocodiles. These are phenomenal answers. Okay, good. I love these answers. So the answer to that question is IDK. Whoever put IDK was correct because we don't know what eats snakes, right? But we have some ideas. And one of those things that eat snakes are birds. So whoever said birds, give yourself a pat on the back. Everybody give yourself a pat on the back. But whoever said birds was onto something here. So I really like birds of prey. I'll also call these raptors a lot in this chat. I'm not talking about velociraptors. But birds of prey or raptors love snakes. They love to eat snakes. Here you can see a red-tailed hawk carrying away a rattlesnake. So let's think a little bit about raptors. So raptors are what we call an opportunist. And all that means is that they will eat whatever walks by them, right? So if you're a venomous snake walking in front of a raptor, you are on the menu. So most raptors eat things like rodents, mice, birds, all that jazz. But at the end of the day, there's a ton of raptors that will eat venomous snakes. And there's a couple pictures of that here, right? So the first two are red-tailed hawks. The one on the left is eating a prairie rattlesnake. The one on the right is kind of grabbing a timber rattlesnake. But this is pretty widespread. And the picture on the far right here shows an Asian bird of prey, an Asian raptor. And this is a crested serpent eagle. And I know you can't really see what he's holding in his claws there, but he's holding a cobra. Cobra is a horribly deadly snake. And whoever wanted to see cobras, here's another cobra. So again, this is really widespread. Raptors and venomous snakes are kind of a really pervasive idea even in human culture. So the image here is off of the Mexican flag. We've got a snake fighting with an eagle. So people have spent some time thinking about this, at least a little bit of time. And one thing that kind of gets me really interested is this idea of a snake specialist birds of prey. So I don't know if anyone knows what a secretary bird is. It's got these beautiful long legs and a cool kind of plumage on its head. They love snakes. We've got birds called seriamas, snake eagles, laughing falcons. And these are raptors that eat a ton of snakes. They love snakes. So this is certainly something that should be played with. So how do raptors avoid snake bite? Well, they could do it through physical mechanisms, right? So maybe they're just built in a way that makes them pretty resistant. They could do it behaviorally. So maybe they just know how to work with a snake. Maybe they know how to not get bit. Or they could do it physiologically, right? So there could be something inside of the blood of that raptor that actually helps in the case of an envenomation. And that's what I wanted to play with. So I did just that. So again, venom, we know about this, we know about this. But what do we know about venom and predators, right? So shifting the focus from squirrels and mice and prey items, what do we know about predators? Well, the honey badger, right? Everybody knows the honey badger don't care. The honey badger is very resistant to the venom of things like this Egyptian cobra or this Cape cobra. It's very good at inhibiting neurotoxins. Same type of thing with the Virginia opossum and with possums broadly. They can really detoxify venoms from North American rattlesnakes. And then a really cool case is the mongoose, the Egyptian mongoose. And he can actually turn off these things called seraphotoxins that are horrible. They just melt prey items. So really cool. But then there's examples where we cannot inhibit toxin. So we talked about our ground squirrel, we'll leave him alone for now. But peafowl, peacocks love to eat snakes. And this study found that peacocks were not resistant to snake venom, to crate venom. Again, right, that serpent eagle study found that serpent eagles do not have any resistance to cobra toxin. So weird. Why are you going to eat a cobra if a cobra bite could kill you? And that's what kind of got me going, right? So I wanted to look at this at home in North America. And what I did is I said, oh, the Great Plains would be a fun place to play around with this idea. So I took our same prairie rattlesnake that we talked about earlier, and I checked for venom resistance in a bunch of different raptors that live here. So I used the red-tailed hawk, the Swainson's hawk, the bald eagle, the golden eagle, and the great horned owl. And what we did is we didn't inject any raptors with venom because we like raptors and raptors are protected, and that wasn't really needed for what we were doing here. Well, we did do that same test I talked about where we mixed up venom and blood, and we looked at percent inhibition. So for that, we used 9 red-tailed hawks, 1 Swainson's hawk, 3 golden eagles, 3 bald eagles, and 9 great horned owls. And then we needed a control, right? We couldn't really use lab mice because a bird and a mouse might work differently. So we went ahead and used chicken. So we took blood from 3 chickens and used that as a control. We took venom from a single rattlesnake just to avoid any venom variation. And then again, we looked at percent inhibition. So here's exactly what we found. And there's a lot of bars here, but this isn't super important to kind of get bogged down with. The key point here is that chicken is 0, right? We would expect that. We don't think a chicken would have resistance to a rattlesnake. And the rest of these raptors are all pretty close to 0. So that axis goes up to about 30%, right? So keep in mind that's not 100%, but these values are low. So raptors probably aren't resistant to snake venom, is kind of our take home. If we kind of flip those numbers and look at them a different way, let's look at them as actual numbers, right? As percentages. These are average percentages. So chicken, 0%. He cannot turn off snake venom. But in comparison, the rest of these birds are also really, really low. So things like 3%, 5%. If you're turning off 5%, you're probably not shrugging off a snake bite, especially when we think about our rodents earlier, right? Where a pack rat might turn off 80% of that venom. And sometimes he gets by, but he can still also get eaten by a prairie rattlesnake. So raptors across the board, at least in the Great Plains of North America, do not really seem to have anything going on as far as venom resistance. So what does this mean? Like I just said, nothing is going on here. Raptors are sensitive to rattlesnake venoms. And if you dig enough into the literature, you can actually find papers that talk about this. So apparent fatal snake bite in 3 hawks. Myself and a couple colleagues actually just published on this in a bunch of different hawks. So we see kind of across the board, raptors are not really good at getting bitten by snakes, I guess is the gist of it. All right, so what that means is that these birds are probably relying on physical or behavioral adaptations to keep themselves safe. So maybe really, really thick feathers, right? You can think of a hawk having really fluffy plumage. Maybe that just keeps fangs from touching that hawk. Think about the feet of a raptor, right? Their armor. So maybe the fangs can't actually get through, can't really get a purchase on his feet very easily. And then there's also some weird blood flow things going down with raptor feet, bird feet in general, right? There isn't a lot of blood in the foot of a bird. So maybe even if a snake does get venom in, that venom just doesn't really get into the body of the raptor very effectively. So that's an idea. And of course, behavior, right? So this snake eagle on the right here is doing a bit of a dance. A lot of different birds of prey do kind of weird looking dances, if you will, to confuse snakes. And I think maybe that's really, really effective in just avoiding a bite. All right, so future work, right? It never ends. So I talked about those snake specialists a moment ago. What safeguards do these specialist snakes have in place? Is it really just physical and behavioral, or is there something physiological? Is there something happening inside? And this kind of really piques my curiosity. One of these pictures here is a laughing falcon. It's holding a venomous lancehead. This snake kills a ton of people. Yet this bird, who's really no bigger than a robin, is eating one, right? The secretary bird here, that's the one with the beautiful legs I was talking about a moment ago. It's gulping down a puff adder. So I would love to dig into this a little bit more. All right, so with the last kind of remaining few minutes that I've got, I want to segue into what I'm currently curious about. Not that I'm not curious about number 1, but number 2 is kind of what's taking a lot of my focus right now. So this is snake biodiversity, venom, and how it relates back to us as people. So I'm working on this here at Oklahoma State with Dr. Gwyn Wogan, who I kind of gave a shout out to before. I would encourage you to check out her lab group and our work. But again, quote, right? My favorite quote that I've talked about today. So I won't repeat it, but this time I'm going to agree with AF Dutch. I'm going to agree that snakes are kind of not liked, right? And somebody asked me a little bit earlier on about snake detection theory. And I want to kind of push this idea forward. And this is the idea that maybe we, and when I say we, I mean you and I and people, and snakes in general, maybe we've been feuding and interacting and, you know, essentially fighting for a lot longer than we like to think. And there's really a big body of evidence to support this. The book that I've shown here is a great read. I would encourage anyone to read it if they are interested in snake detection theory. But it summarizes a lot of data. And one of those lines of data is a spitting cobra, right? So we know as of relatively recently that spitting probably evolved in tandem at the same time that our early human ancestors began standing up, right? So if you're being chased by a monkey on two legs who's wielding a stick or who's throwing rocks, you need to spray venom at them. So it's fascinating that we did that to snakes, and conversely, snakes did that to us. And then this whole idea of snake recognition, right? So a young primate, whether that's a human primate or a non-human primate, there's a plethora of studies out there that just show this really uncanny ability of baby primates to just pick up on snakes in the environment. They recognize them more than they recognize other things. So I think there's something going on here, but I'll leave that at that for now. All right, so snakebite. So snakebite is a horrible global problem, and it kills 100,000 people yearly. Sounds like a huge number, right? But that isn't actually the problem. That isn't the issue. It's a problem, but it's not the issue. The issue is that snakebite disfigures an additional 400,000 people yearly, right? And you'll think, well, Neil doesn't know what he's talking about. I would rather be missing a finger than be dead, right? And that's a fair argument. But if you live somewhere, particularly in the developing world, where your livelihood and your paycheck, right, comes from working in the field or working with your hands or your feet, or just having a healthy body. If you lose a leg, you lose a hand, you lose some fingers, you're potentially out of work, right? And it's because of this that snakebite actually can drive the cycle of poverty in the developing world. So if you're already in poverty, losing a leg when your life depends on being able to walk around pushes you further and further and further into poverty. So this is a problem. And because of that, folks have started to pay attention, especially recently. And we see here that the World Health Organization has revealed a snakebite envenoming strategy, a global strategy. So they're really paying attention. And then conversely, a bunch of money has become available recently. So this article just shows $100 million boost for better antivenoms. People are paying attention to snakebite, finally. But why? Why is this a problem? Why can't you just go to the hospital? So in the developing world, there seems to be a reliance on traditional healing. So maybe misguided beliefs, people are putting charcoal on a snake bite, or they're trying to cut that snake bite with broken glass, right? And hoping that'll fix things. Of course, that doesn't work. There's also issues with antivenom. So can I get antivenom? Can I afford antivenom? But then the most interesting thing, and this is what kind of gets my gears turning in my brain, is this idea of the lack of efficacious antivenoms. So I get antivenom and they give it to me, but what if it doesn't work? Why wouldn't it work? And this happens a lot. So this is the third time we've seen this. You guys are tired of this. Venom does stuff, but venom is variable, right? So if we think about one example in the US, this is the Mojave rattlesnake. We see 4 different types of venom in this one snake. I mean, what a weird scenario, right? And this snake doesn't even have that big of a range. So we see variation that could come from phylogeny. All that means is genetics. So maybe how related are you to another snake? We see variation that could come from the environment and the climate. So maybe temperature, precipitation, all of that is affecting venom. But then we see variation from diet, right? So you are what you eat type of idea. So venoms are super variable. And that kind of gets me to this idea of the central dogma of biology. So I know a lot of you guys are not too deep into biology yet. You're still kind of getting there on biology. But we talk about this a lot. So DNA, RNA, protein, we have a linear workflow. Things move in a line. And I think there's a similar idea with snakebite, right? I think things move in a line. And I think we can look at that starting at diversification. So what I mean by that, again, are these genetic relationships. How are snakes related? We can move up the line and look at how venom varies. And then finally, we can look at antivenom. And we can do that in one snake, which I am doing, and that's a monocled cobra. So a snake we're very familiar with. Everybody loves cobras, but we don't actually know much about them. So you can see this guy here playing with cobras at a roadside attraction. You can see these captive cobras. You can get cobras really in any color, but we don't know much about cobras, right? They have a huge range here from India all the way through Southeast Asia. And in that range, we know nothing. So let's start at diversification. So a lot of the work that kind of described cobra diversity happened in the 90s. And this used specimens. It used dead cobras. And these dead cobras are great, right? A lot of the time they look just like a live cobra. Sometimes they don't look like a live cobra. This is a pair of boots that's in the Paris Museum of Natural History. So again, your mileage may vary if you're describing cobras based off of these specimens. So what we could do is we could look at genetics, and this has become really big lately. And this monocled cobra, when we've looked at it genetically, we actually find that there's several different genetic lineages. And these names are all Latin, so they're not very helpful for you. But just pay attention to my brackets here. So we've got a lineage in Bangladesh, And we have a lineage in Burma and Thailand that seem to be very separate genetically. Someone looked at this again a little bit more recently, and they found a better resolution here. We have a couple different lineages. We have 4 different genetic lineages in this one cobra. And then most recently, we have 2 really well-supported lineages, kind of in this red box. So maybe 2 species going on. And then just a few weeks ago, this is brand new, somebody actually described a new species of cobra from the monocled cobra. So this snake, which looks very different, right? It has bands on it. It's got a little bit different of a pattern. This is a new species of cobra that has been hiding from science forever, just because people haven't looked. So I think these snakes look different. And I think there's probably value in digging into this a little bit deeper. So the monocled cobra may actually be the monocled cobras. Maybe they're species hiding right in front of us. All right, venom variation. We talked about that a minute ago, so same figure. Monocled cobra venom is very variable, and it's variable even within, across populations. So venom in Malaysia, venom in Thailand, venom in Vietnam are different. They're all different. But it gets more complicated because even in one population, venom can be different. So these are 2 snakes from India. Don't worry too much about the words or the abbreviations, just worry about the colors. And you can see that those charts, those donut charts, the colors are all over the map. Venom is very variable from one snake to another. All right, so how does this variation in venom, how does that correspond to genetics? We don't know. I want to know. Then finally, antivenom and clinical management. So antivenom is the only real treatment against snake venom that works, right? So charcoal doesn't work, broken glass doesn't do anything. We need antivenom. And antivenom is basically made when we inject a horse or a sheep with a little bit of venom, and it builds up immunity, and then we kind of purify that and we take that. But what happens if the venom of the snake that bites me and the venom that my antivenom was made with, what if those are different? And we do see that. And one example of that is that Indian cobra venom, so snakes from India, and then snakes from Sri Lanka, if we mix these antivenoms, they don't work. They just plain don't work. So if you're bitten by a snake in Sri Lanka and the only cobra antivenom comes from India, you're maybe going to die even if you get antivenom. So this is a huge problem. Same thing with our monocled cobra, right? So if you're bitten by a monocled cobra in India, and for whatever reason, the Thai antivenom produced from snakes in Thailand is what you have available, this probably isn't going to help you very much. So this is really important. All right, so then the question is, what other gaps are present in this antivenom coverage? If I'm bitten by a snake, let's say in China, and maybe I have a Malaysian antivenom, will that work? No one's ever looked at it. We don't know. All right, so I kind of just want to fix snake bite. That's kind of the goal here. And I think doing all of this, right, building from the ground up is a really great way of getting there. So we're kind of creeping up on the end of time here, and I'm just going to leave you with this kind of idea. I want to impress this idea upon you that not knowing stuff is one of the coolest things, right? I think it's cooler than knowing stuff. And a couple examples of just things that keep me up at night. So garter snakes, right? Kind of a snake we're all used to. We have an island where I come from. It's 3 miles off the shore in the middle of a lake. In the spring and the fall, garter snakes swim from the island to the mainland and then back from the mainland to the island. How are they doing it? We don't know. I want to know. Another example, this little snake comes from Somalia on the coast. We have no idea what it is. We think it's a puff adder, but no one's ever looked at it. And I want to look at it and know. 2 more examples, right? Burmese python. I could tell you everything about this snake in Florida, but the snake doesn't come from Florida. It comes from Southeast Asia. We have no idea how it lives in Southeast Asia. And then, of course, this is a really cool picture. This is a snake eating a tarantula. This is a group of snakes called Stenorrhina. I'm not sure if they have a better name than that, but these snakes eat tarantulas. That's what they eat. That's a scary thing to eat. How are you eating a tarantula? Anyway, I want to know. And I think the big point is that if we all took a moment out of our day or out of our week or whatever, and just appreciated a snake, right? Or appreciated the natural world, whatever your thing is, I think that would be step 1 to answering some of these what we don't knows. So I would encourage you, right? Next time you're outside, flip a rock, flip a log. There might be a little snaky friend hiding underneath who can maybe teach you something about snakes. So that's all I've got. Thank you so much for kind of being attentive and being here for this. And at this point, I'd be happy to take any questions that you guys have. Thank you so much.
Hello.
All right, so I'm gonna
Okay.
open up the Q&A. If anyone has like verbal questions, why don't you go ahead and just ask me, and then we can go ahead and flip to Q&A maybe.
Good question. They want to see a snake.
They want to see snake.
We're
I
all
wish,
silently standing behind the cameras, like chanting snake, and we were wondering if you had any that you could like show us.
I wish I did. Unfortunately, I'm in the lab right now, so the only thing I could show you a dead snake actually. Would that be okay?
Yes.
Yeah, sure.
Yes.
Go ahead.
So we talked about specimens a moment ago, right? Oh gosh, this bucket is tight. All right, this is gonna make my hands dirty, but I'm gonna do it for you guys. So we talked about specimens a moment ago, right? So specimens are really important to science. We can learn a lot from dead snakes. So this hopefully won't drip too much, but this is a cobra. This is a dead monocled cobra. It's from a museum. And this animal is super useful to researchers to learn things from it. So we have a bucket of these here. Unfortunately, I'm not at home. If I was at home, I could pull out a huge python and we could do some python time. But right now we're gonna have to settle with this alcohol kind of pickled monocled cobra for the time being. So I apologize, but maybe next time if you guys have me come back, I'll plan this better.
All right.
Perfect.
Right. Is that question? All right. So let's see the questions here.
Most of the questions were asked by the elementary kids. They all left.
Yeah.
School ended. So
If you guys have any,
there's still a lot
I
of
don't
people here.
I don't know if you guys want to pick questions, or I can just go ahead and scroll through them. But I'm happy to.
We can pick them. So here's an interesting question. So, how many of the 4,000 species are poisonous?
That's a really good question, and the best answer I can give you is I don't know. And the reason I give you that answer is because venomous snakes, for the longest time, right? We only ever considered snakes to be venomous if they could hurt people. So that means things like rattlesnakes and other vipers and cobras. But that snake that I showed you eating the tarantula, that is a venomous snake. It has venom, but its venom only works on tarantulas. So there's a lot of snakes in the world that have venoms, and they have very potent venoms, but their venoms are made for things like tarantulas, right? Or things like mice. So how many snakes are actually venomous? I don't have a good question, or I don't have a good answer for that. But that's something that I think someone needs to work on badly. One of you guys, maybe, if you catch the bug for snakes, should look into that.
I have a movie question. Do you like studying structure and function or cause and effect more?
That is a great question, and I don't know if I have a good answer for that. So the answer that I always tell people when they ask me what I like to study, I guess in particular, I'm a snake guy. So at the end of the day, my questions come back to how does a snake live in the world? How does it interact with the world, right? So sometimes that does take me in a more structure and function way. I want to know how things are built, how things work out and interact with each other. But sometimes it can also be cause and effect, right? So I think I do both would be my answer to you guys.
Good answer.
What?
Okay, so I also have a question, which is, was there like a moment where you were like, yes, snakes are the thing I want to do, or did you just like always know that, yep, I'm gonna do snakes when I grow up or something?
That's a great question. I think I probably like laid in the crib as a baby just with snakes floating around in my mind. I don't think there was ever a pivotal moment or anything like that. But no, I was always a weird kid, you know, while people were playing soccer, I'd be catching bugs and thinking about everything else. So I think snakes were always in there. That's what I'm gonna go with.
What is your least favorite part of working with snakes as a scientist, if any?
My least favorite part. This is a great so I have two least favorite parts. My first least favorite part is getting pooped on. We get pooped on a lot in science, in organismal science. So getting pooped on is not fun. It's okay, but if you don't have a change of clothes, it's kind of yucky. The thing that kind of is unfortunate, right, is that we sometimes do have to kill snakes to learn about snakes. And this is something that we grapple with a lot, right? And, you know, if you love snakes, you don't ever want to hurt a snake or even say a mean thing to a snake. But at the end of the day, right, like sometimes you do need to kill a snake to really understand how a snake works. And you just have to remember that like the end goal is to do good, right? And if that means, sacrificing an animal somewhere along the line, you just hope that you're really giving back as much as you can.
This is also kind of a dumb question unless you had one.
I was just gonna read one.
No, we had a dumb question of from Amber, she's sitting next to me. What is your least favorite snake? And follow-up question, what's your favorite snake?
Okay, awesome. Great question. So there are no dumb questions, first of all. So I'm gonna twist your question a little bit, because my least favorite snake is actually a lizard. And it's this critter that comes from Greece. It has a weird European name. It's called a sheltopusik. You could also call it just like a European legless lizard, but it looks like a snake. It looks very similar to a snake, and it may be my least favorite animal on the planet. I'm gonna go and say that because it poops everywhere, and it only eats snails in the wild. So the poop has this really distinctive odor, and it kind of lingers on you forever, and it's just kind of a gross creature. So I respect them, but I don't like them is the answer I'm gonna say.
All
And
of
then
the biology nerds are nodding their heads along with you.
Yeah, and then my most favorite snake, I'm a cobra guy. I love cobras, so cobras are the move. Yeah.
So here's just a question that I read off this. What is the smallest snake or smallest known species of snake?
Great question. So I don't have a species off the top of my head for you, but it'll be a creature called a blind snake. So it's this, again, diverse radiation of snakes, and they look like earthworms. They're itty bitty. And there's one of them that's super common. It's called the Brahminy blind snake. They also call it the flower pot snake. And it's a cool snake. It doesn't need to mate. It's what we call asexual. So a single one can make more young. And they've traveled all around the world through the garden trade. They live in flower pots. So if you ship plants to Hawaii, for example, there could be a blind snake hiding in a flower pot, and it will breed. It doesn't need a mate. So really cool.
I'm wondering your feelings about failure. Like what role does failure play in your work?
That's a great question. I love that question. So I fail more than I succeed, guaranteed. I feel like everything I do is failure. And a lot of this, and a lot of science, right, is just screwing up again and again and again until you get it right. So I think without failure, this would all be too easy and we wouldn't be motivated to do it. So that's kind of the stance that I'm going with. But I think failure is important, and it pushes us to want things even more and to want to work to figure them out. So I'm a big fan of failure. I think it's important.
All right. So there are a couple questions that have also asked about, potentially being poisons, if you were to say consume or lick a snake. So maybe could you maybe explain the difference? Like, does that happen? And maybe the difference between poison and venom. Just
Yeah. So this is a can of worms, and I've got a lab mate, actually, another PhD student here, who gets really mad about this. If you call a snake poisonous, he will kind of blow up into a little panic attack. So the thing is, right, poison and venom are actually two different things. So poison is any toxin, any toxic thing that you can consume or you can inhale or you can rub on your skin, and that will make it toxic, right? Venom, on the other hand, needs to be injected. So venom always comes from a creature that needs to poke you or bite you or something like that. So the running, I don't know if it's really even a joke, but you can generally lick a snake and you're okay, right? But a snake can't lick you or bite you. That would be bad. But we're going to make it even more complicated. There's a couple of snakes out there that are also poisonous. So there's this group of snakes in Asia. They're called keelbacks, and they eat a lot of poisonous things. They eat toads and frogs that have poison. And what they do is that they take that poison from the toad or the frog, and they actually store it in these big glands on the top of their neck. So if you caught a keelback is also venomous. So a keelback could bite you and make you hurt a little bit. But if you lick that keelback, you could also get really, really sick. So in general, snakes are venomous, but poisonous snakes are also out there. So be aware, know your definitions. Yeah.
All right, I think we're gonna wrap up.
yeah.
Perfect. Awesome.
Right?
Okay, well, it was great. Thanks for having me, guys. It was a lot of fun talking to
Thank you so much for coming.
Yes,
Yeah,
thank
for
you.
presenting.
you. Cool. Thanks.
Okay,
Before we go, though, we do have
we have a
one
quick
announcement to
announcement
make.
for all participants. So the National Biodiversity Teach-In is a student project that has some partners, and one of our partners is Earth Echo International. And right now they're doing the Our Echo Challenge to empower young problem solvers in STEM. And there's a competition going on for grades 5 through 9. And if you guys want more information about the contest and prizes involved, go ahead and email us. We'll get it to you. Thank you again.